Hello everyone. In this series of the videos, we are going to discuss about interesting topic of molecular biology, that is transposons, also known as mobile DNA element, jumping gene, insertion sequence, or the cassettes. In this lecture, I'm going to focus on history or the discovery of transposome by Barbara McClintock, then transposomes which is present in the animal like Drosophila, plant like maize, and some transposome which is reported in the bacteria. Then I'm also going to discuss the different type of transposome, mainly DNA transposomes and retrotransposones, and their mechanism of transposition, mainly replicative or copy paste mechanism and non-replicative or cut paste mechanism. In the later half of the video, I'm going to focus on important type of a transposable element, that is P element, which is present in the Drosophila. Okay, I'm going to discuss in detail the disorder which is caused by this P element in the Drosophila, known as hybrid dysgenesis. Okay, before starting with the actual discussion, we have to honor or we have to remember Barbara McClintock for her work in the cytogenetics, because she is the person behind the discovery of transposable element. Actually, she was born in 1902 and she started her college in Cornell College of Agriculture in 1990, and then she completed her BSc in 1923, her MS in 1925, and her PhD in the botany in 1927. After completing her PhD, Barbara developed interest in the maize plant, and then she focused on the cytogenetic study of the maize plant. In her study in 1931, she first time reported the complete genetic map of the maize plant. Then during her work, she also developed a technique for observing maize chromosome, and she also reported the structure of one of the maize chromosome. In 1938, she able to find the Neurospora crassa life cycle. In 1944, okay, she started the famous experiment of her on the maize plant, which finally end with discovery of the transposons. Okay, in 1944, she started the study in which she find that the maize plant have three different type of color pattern in the seeds. Some maize seed show purple kernel color, some seeds show spotted color, while some seeds are colorless. And finally, she concluded in 1948 that this difference in the color pattern of the maize seed is generally observed due to two different type of element which is present in the maize genome. She named as DS and AC, that is dissociation and activator. And both these elements are nothing but the transposable elements or transposons. They have ability to change their position from one side of the genome to the other side. Okay, and due to these two genes, different type of color occur in the maize seed. She retired officially from her work in 1967, but world know the importance of Barbara and her work only after F. Jacob and J. Monod first time reported the lac operon in Escherichia coli. Okay, because this concept before them was already told by Barbara McClintock. According to her, there are some genes which is present in the DNA which having ability to regulate the genome, which having ability to change the position from one side to another side. These genes can easily travel. Okay, and due to her work, at the end in 1983, she received Nobel Prize in Physiology or Medicine. She died in 1992. Okay, so one cannot start the study of transposon before remembering this great scientist Barbara McClintock. Okay, now the definition of the transposome is that the sequences which is present on the DNA or on the RNA, which having ability to move from one side to another side on the same chromosome or on the different chromosome. Actually, these are the sequences which can be classified into two types. That means transposomes are broadly classified into two types as autonomous transposomes and non-autonomous transposomes. That means some transposome having ability to move from one side of the DNA to the another side by using enzymes coded by the transposome itself. That means this type of transposome do not need help of any other enzymes or any other protein from other gene. They already have the enzyme like transposase for their transfer. So they are called as autonomous, while non-autonomous transposomes generally need help of autonomous transposomes. Enzyme produced by the autonomous transposomes used by non-autonomous transposomes for translocation. The process by which transposome change their position is called transposition. Okay, now in this lecture, I'm going to mainly focus on the P element of Drosophila, but actually Drosophila is the model organism. We know that in the genetics study, 15% of the genome of the Drosophila is reported to have transposable elements. Okay, that means the organism may contain 15, 30, 35 or 50% genome which is made up of transposable elements. Then in maize, approximately 80% genome is showing transposable property, while in human 45% DNA show transposable elements. Then actually, two type of transposable element in the maize is reported by Barbara McClintock as AC and DS. In my upcoming video of the series, I am going to discuss about transposable element in maize. Okay. Then bacterial transposomes, generally prokaryotic organism like bacteria have DNA transposomes. That means the transposomes is made up of DNA and it can be transferred from one site to another site in the form of DNA. Okay, an example of bacterial transposomes are like insertion sequence or composite transposomes and sometimes retrotransposomes. Okay, then the important type of transposome which is generally present in the eukaryotic cell is retrotransposome. Okay, now there are two different type of transposomes according to their mode of transfer, like DNA transposomes and retrotransposome. What is the major difference between these two transposomes? DNA transposome can migrate from donor DNA to the recipient DNA directly in the form of deoxyribose nucleic acid. Okay, that means during the transfer process, DNA is excited from the donor DNA and then it is inserted into the target DNA in the form of DNA only. So it is called as DNA transposomes. While retrotransposomes use different way for transfer. Actually, retrotransposomes are the type of transposomes which first transcribe, that means DNA convert into RNA by using RNA polymerase, and the RNA copy is again converted into DNA, complementary DNA, by reverse transcriptase, and now this new copy of DNA insert into the target site. So it is called as retrotransposition. So these two type of transposomes are mainly present, out of which DNA transposomes mainly present in the bacteria, while animals and plant generally show retrotype transposomes, like human show lines and shines. I'm going to discuss about transposomes which is present in the human in my upcoming videos. The mechanism of transposition. Transposomes use two different way for their movement, like replicative transposition and non-replicative transposition. In case of replicative transposition, transposomes replicate in the donor site so that exact copy of the transposome or the gene is produced, and the newly synthesized copy migrate and inserted into the target site. That means in this case, transposome going to increase its copy number. Original transposome remain as it is in the donor DNA, while new copy inserted into the target DNA. So this type of transposome is called copy-paste mechanism, because the copy number of the transposome going to increase, and the mechanism is similar to that of the replication that occur in number of different type of organisms. Then non-replicative transposition is a type of a transposition in which transposomes, original copy of the transposomes, get excised from the target or donor DNA, and then it get inserted into the target DNA. That means in this case, copy number of the transposome do not increase, only the site or the position of the transposome, original transposome, change from donor site to the target site. So this type of mechanism is called as cut-paste mechanism. In this type of transposition, replication of the transposome do not occur, copy number do not increase. Okay, so this is all about the general introduction of the transposomes. Remember one thing, transposomes are very important. We have to study them carefully because number of different type of genetic disorder and number of different type of cancers in the human is caused by transposons or mobile DNA element or jumping gene. Now in this lecture, I'm going to focus on this point, that is transposons in Drosophila melanogaster. We know that this is the ideal organism for genetic study. Number of different type of genetic experiment was carried out on the Drosophila. So they have found in 1967 two different type of transposable element in the Drosophila, mainly P element and Copia element. Both these are the type of transposomes which is present in the genome of Drosophila. In this lecture, I am going to focus on P element, but before that, I just want to give quick introduction of Copia element. Copia element are nothing but the sequence of the nucleotide, a gene which is present in the Drosophila genome. It is a 5 kilo base pair, 5000 base pair sequence. Multiple copies of Copia element is generally present in the Drosophila genome. Now here you can see total length of the Copia element in the DNA is 5 kilo base pair. Both the site of Copia element have 276 base pair long terminal repeat. Here you can see 276 base pair long terminal repeat on both the site of the transposomes. And at the end of the transposomes, outside the long terminal repeat, 17 base pair inverted repeats are present, IR. 17 base pair inverted repeat is present outside the LTR region. Okay, so main length or total length of the Copia element in the Drosophila is 5 kilo base pair, and at the both end of this gene, 276 base pair long terminal or LTR regions are present, and outside the LTR in both the direction, 17 base pair inverted repeats are present. These 17 base pair inverted repeats play important role at the time of transfer of this gene from donor to target. Okay, now at the time of transfer of this sequence, actually first the DNA is converted into RNA by the process of transcription. We know that this process is catalyzed by RNA polymerase enzyme. And after formation of the RNA, now reverse transcriptase enzyme convert the RNA into complementary DNA. Okay, so this is the type of retrotransposones. And once this DNA is produced, now it is ready to enter at a new site in the genome. So this is the mechanism by which this Copia element transfer themselves from one site of the Drosophila genome to the other site. They have the ability to jump, so they are called as jumping genes. Now I am coming towards main point of today's lecture, that is P element in Drosophila. Okay, so this is the structure of P element which is present in the Drosophila genome. Total length of the P element is 2.9 kilo base pair, that is 2900 base pairs. Okay, both the end of the P element having 31 base pair inverted repeat. Okay, here you can see 31 base pair inverted repeat present at both end of the P element, and actual protein coding region of the P element have 4 exon, okay, and only 3 introns. Remember this thing, the protein coding region of the P element have 4 exon, and in between the 4 exon we know that non-coding regions are present, they are introns. Okay, so this is the general structure of P element in the Drosophila. Now after understanding this structure, we are going to see the condition that occur in the Drosophila due to this P element. Okay, and the disease or the abnormal condition that occur in the Drosophila due to entry of this P element called as hybrid dysgenesis. What is hybrid dysgenesis? It is the condition in which the progeny of the offspring of the Drosophila show frequent mutation in their genome, chromosomes show breakage, and the offspring shows sterility. That means after development of hybrid dysgenesis, the progeny of the Drosophila cannot develop the fertile gametes. They cannot show the fertilization. Okay, so they are sterile. So three important characteristic feature of hybrid dysgenesis is that frequent mutation or high rate of mutation, then breakage of the chromosome, and sterility. Okay, so these three are the characteristic feature of hybrid dysgenesis condition. Okay, now this is the mechanism by which you can understand how exactly this P element cause hybrid dysgenesis in the Drosophila. Before that, we have to know that in Drosophila there are two different type of strains are present. They are M strain and P strain. Okay, M strains are nothing but the type of a Drosophila in which P elements are missing. These elements are missing in the genome of this M strain. Okay, while the P strain always have P element or this transposable element in their genome. This is the difference between M strain and P strain of the Drosophila. Now, by observing this Punnett square, you can easily understand if female is M type, okay, female is M type and male is M type. In that case, the offspring or F1 generation always show normal characters. They do not develop this syndrome, hybrid dysgenesis. Okay, number one case. Number two, if the female is P type, next type, okay, and male is M type, after fertilization, the F1 generation is again normal. They do not show hybrid dysgenesis. And in the third case, if the female is P and male is P, in that case, the F1 generation is normal. So, these three crosses, M female with M male, P female with M male, and P female with the P male, produce normal offspring. They do not show a hybrid dysgenesis. But M female, when crossed with P male, this is the only condition required for hybrid dysgenesis. If the female of M strain cross with male of P strain, in that case, it will going to develop the disease which is hybrid dysgenesis. I am going to focus on this condition, the cross between M female and P male. Okay. Now, this is the condition in which female is P type and male is M type. Okay, and the another cross in which female is M type and male is P type. So now, if female having P element, okay, and male do not show P element, that means male is M type, and if we cross these two, then during the F1 generation, all the offspring are normal. Okay, they do not show mutation in the chromosome breakage, or flies do not show sterility. They are fertile. Okay, that means this hybrid dysgenesis not occur in this cross. Okay, you can take instead of this any one of this cross, three cross. We're going to get similar result. Okay, but now if female is M type, okay, and male is P type, that means female do not have P element, but male have P element. Now, in this case, when fertilization occur, the sperm will going to carry the P element into the egg of the female of M strain, and after entry into the germ cell of the female, okay, this P element from male will going to get activated, and after activation, this P element cause hybrid dysgenesis in F1 generation, which is not good for the progeny of these two parents. Okay, and this happened because the P element which is present in the male get entry into the egg of M female, and this element only get activated after entry into the egg of M female. This is very simple concept. Okay, the complex thing is that two questions that always puzzle the scientist. Number one, why hybrid dysgenesis only observed in germ cell. Okay, as I told earlier, the P element from the male get activated only when it get enter into the germline cell of female, that is egg of female. Okay, this P element do not show any action in the somatic cell. We know that animal body is made up of two different type of cell, germ cell and or gamete producing cell and somatic cells. Okay, and this element, this condition only occur due to action of the P element in the germline cell. So the question number 1 that arise in everyone's mind is that why hybrid dysgenesis not observing, only observed in germ cell and not observed in somatic cell. One of the best explanation for this thing is that hybrid dysgenesis occur only in this case when P element from the male enter into the egg of EM strain female. Okay, and inside the germline cell, when this P element occur, we know that this is DNA. So once it converted into RNA, okay, after conversion of RNA, formation of the RNA, the splicing of the RNA occur in which these introns are removed and these exons are joined together to form mature RNA. Okay, and the process of splicing is correctly occur in the germ cell. So mature RNA from this P element is only produced in the germ cell and get activated. But in case of somatic cell, if the P element enter in the somatic cell of the female, in that case splicing cannot occur properly and one of the intron is not removed from the RNA at the time of splicing. This is the mistake that occur by the splicing machinery inside the somatic cell. And due to this mistake, one of the intron remain as it is in the mature RNA, and due to that intron, mature RNA cannot get activated after translation. The proteins which produce cannot develop hybrid dysgenesis, and due to which somatic cell remain away from this condition. This condition only occur in the germ cell. Okay, so it is called as hybrid dysgenesis is a germline phenomenon. Okay, so this is the most accurate answer for this question. Another question which come in number of competitive exams, like why offspring of P female and P male or P female or M male, okay, these conditions are not showing dysgenesis. Why they are not showing dysgenesis? Number 1, number 2, number 3. Okay, the best explanation for this question is that though in this cross both P female and P male have P element, this element, still they do not show hybrid dysgenesis. This is because if female having P element naturally in her body, in that case female also have machines or mechanism to avoid the harmful effect of the P elements. Okay, in that case female body have or female germline cell have special type of RNA and a protein called PI protein. The combination of the RNA and PI protein occur inside the P female. Okay, and now this PI mRNA complex will going to suppress the effect of P element inside the P cell. If in this cross, this P element from male enter into the egg of the P female, in that case P female egg always have the solution, that is PI mRNA complex, and that complex will going to inhibit or going to suppress the P element from female as well as P element action from male, and due to which the progeny which is produced by the cross of P female and P male are always normal. They do not develop. P female and P male are always normal. They do not develop hybrid dysgenesis because P female always have solution, and the solution is PI mRNA complex. Okay. Now in this case too, as female is P female and M male, P female and M male, all progeny are normal because female is P. In that case, female's body have PI mRNA complex, and that complex will going to suppress the P element which is present in the female, which is not going to activate it, and due to which the progeny of these parents, P female and M male, will going to always remain normal. And no question for this cross, as both M female and M male they will going to develop normal progeny because they do not have P elements. So this is all about the transposons which is present in the Drosophila. In today's lecture, we see the brief history of transposons or mobile DNA element or jumping gene discovery by Barbara McClintock. Then we discuss in briefly about the transposome in Drosophila, transposome in maize, bacterial transposome, then different type of transposome like DNA transposome and retrotransposomes, and mechanism of transposition, that is replicative and non-replicative transposome. Then we focus on the transposome which is present in the Drosophila. Number one, Copia element, which is the example of retrotransposome, and P element in Drosophila, which is responsible for hybrid dysgenesis. In my upcoming videos, I am going to discuss about transposons in maize. Thank you.